You're listening to LeadAI.dev, your daily updates on AI development. Today's topic is MolMem. AI memory augmented drug discovery cuts Oracle calls by 60%. Here's a quick overview. New memory augmented AI system MolMem achieves sample efficient molecular optimization, reducing expensive Oracle evaluations by up to 60% in drug discovery workflows. Now, let's ask our analyst about these developments. What's new? MolMem Memory Augmented Molecular Optimization System?
Researchers have introduced MolMem, a breakthrough memory-augmented agentic reinforcement learning system that dramatically improves sample efficiency in molecular optimization for drug discovery. The system addresses a critical bottleneck in pharmaceutical research where each oracle evaluation, the process of testing molecular properties, costs significant time and computational resources. Traditional trial and error approaches require hundreds or thousands of oracle calls to refine lead compounds, while MolMem achieves comparable results with 60% fewer evaluations by leveraging sophisticated memory mechanisms and agentic decision making. The core innovation lies in MolMem's dual memory architecture that combines episodic memory for storing successful molecular transformations with semantic memory for capturing general optimization patterns. This memory augmented approach enables the system to learn from previous molecular modifications and apply that knowledge to new optimization tasks without repeating expensive evaluations. The agentic component uses reinforcement learning to make strategic decisions about which molecular modifications to explore, guided by historical success patterns stored in memory rather than random exploration. Unlike existing molecular optimization methods that either rely heavily on external databases or perform exhaustive search, MolMem creates a self-improving system that becomes more efficient over time. The system maintains structural similarity to original lead compounds while systematically improving target properties like binding affinity, toxicity profiles, or bioavailability. This represents a significant advancement over current approaches that often sacrifice either efficiency or molecular diversity in the optimization process. Reduces oracle evaluation calls by 60% compared to traditional molecular optimization methods, implements dual memory architecture with episodic and semantic memory components for molecular transformations, uses agentic reinforcement learning to make strategic molecular modification decisions, maintains structural similarity to lead compounds while optimizing multiple molecular properties, achieves sample efficiency through memory-guided exploration rather than random search patterns.
Thank you for that analysis. Let's move on. Who benefits from MolMem Memory Augmented Drug Discovery?
Pharmaceutical companies and biotechnology firms conducting early stage drug discovery will see immediate benefits from MolMem's sample efficient approach. Research teams working with limited computational budgets or expensive Oracle evaluations can now explore larger molecular spaces without proportional increases in evaluation costs. Academic drug discovery labs, particularly those without access to massive computational clusters, can leverage MolMem to compete with larger organizations by maximizing the value of each molecular evaluation. The system particularly benefits teams working on novel target classes where existing molecular templates provide limited guidance. Computational chemists and medicinal chemists working on lead optimization campaigns represent another key beneficiary group. These professionals can use MolMem to systematically explore molecular modifications while maintaining detailed memory of successful transformations for future projects. Contract research organizations, CROs offering molecular optimization services can differentiate their offerings by providing more efficient optimization cycles, reducing project timelines and costs for clients. AI researchers developing molecular design tools can integrate MolMem's memory mechanisms into existing workflows to improve sample efficiency. Teams should consider waiting if they primarily work with well-established molecular scaffolds where extensive prior knowledge exists, as traditional template-based approaches may still provide adequate efficiency. Organizations with unlimited computational budgets might not see proportional benefits from the sample efficiency gains, though the memory mechanisms still provide valuable optimization insights. Early stage researchers without established molecular evaluation pipelines should first implement basic optimization frameworks before adopting memory augmented approaches. Pharmaceutical companies with limited Oracle evaluation budgets for early stage discovery, academic drug discovery labs seeking competitive advantages without massive computational resources, computational chemists working on lead optimization campaigns across multiple projects, contract research organizations offering efficient molecular optimization services to clients.
Great insight. Really helpful context. How to get started. Implementing MolMem step by step.
Implementation begins with establishing a molecular evaluation pipeline that can interface with MolMem's Oracle system. Teams need access to molecular property prediction tools, either through commercial software like Schrödinger or open source alternatives like RDKit combined with machine learning models. The system requires Python 3.8 plus with PyTorch for the reinforcement learning components and specialized chemistry libraries for molecular manipulation. Initial setup involves configuring the dual memory architecture parameters, including memory capacity limits, similarity thresholds for episodic retrieval, and semantic clustering parameters for pattern recognition. Configure the episodic memory system by defining molecular transformation templates and success criteria for storing optimization episodes. Set similarity thresholds between 0.7 and 0.9 for molecular fingerprint comparisons to balance memory specificity with generalization. Initialize the semantic memory with clustering parameters that group similar molecular modification patterns, typically using Tanimoto similarity scores above 0.8 for tight clusters. Define the reinforcement learning reward structure that balances property improvement against structural similarity, commonly using weighted combinations of target property scores and molecular similarity metrics. Begin optimization runs with small molecular datasets 5-0. 1-0-0 compounds to calibrate memory parameters and reward functions. Monitor episodic memory utilization to ensure diverse molecular transformations are being stored without memory overflow. Track semantic memory cluster formation to verify that meaningful optimization patterns are being captured across different molecular scaffolds. Validate optimization performance by comparing Oracle evaluation efficiency against baseline methods, targeting 4-0. 6-0% reduction in evaluation calls while maintaining comparable property improvement outcomes. Install Python 3.8 plus with PyTorch, RDKit, and molecular property prediction tools configure dual memory architecture with similarity thresholds between 0.7 and 0.9 for molecular comparisons, initialize episodic memory with molecular transformation templates and success criteria, set up reinforcement learning reward functions balancing property improvement and structural similarity calibrate system with 5-0. 1-0-0 compound datasets before scaling to larger molecular libraries.
That's a very interesting point. Tell us about competitive context. MolMem vs. traditional molecular optimization.
MolMem distinguishes itself from graph-based molecular generation methods like GraphMol and GCPN by incorporating explicit memory mechanisms that capture optimization history. While GraphMol excels at generating diverse molecular structures, it lacks the sample efficiency that MolMem achieves through memory-guided exploration. GCPN uses policy gradient methods for molecular optimization, but requires extensive Oracle evaluations to learn effective policies, whereas MolMem's memory system provides immediate access to successful optimization patterns. Compared to reinforcement learning approaches like Reinvent, MolMem's agentic decision-making reduces random exploration phases that consume Oracle budget without providing optimization insights. The system's primary advantage over template-based methods like Bricks and Recap lies in its ability to discover novel molecular transformations beyond predefined fragment libraries. Traditional template approaches rely on existing molecular knowledge, but struggle with novel target classes or unusual molecular scaffolds. MolMem's semantic memory captures general optimization principles that transfer across different molecular contexts, providing broader applicability than rigid template systems. Against genetic algorithm approaches like NSGA, for molecular optimization, MolMem's memory-guided search proves more efficient than population-based random mutations. Key limitations include the initial learning phase where memory systems require sufficient molecular examples to establish effective patterns. The system's performance depends on the quality of Oracle evaluations, potentially amplifying errors in property prediction models. Memory storage requirements scale with molecular library size, potentially creating computational bottlenecks for extremely large datasets. The agentic decision-making components may occasionally prioritize memory-based suggestions over potentially beneficial random explorations, though this trade-off generally favors sample efficiency. Outperforms GraphMol and GCPN in sample efficiency through memory-guided molecular exploration, surpasses template-based methods like Bricks by discovering novel molecular transformations, achieves better Oracle efficiency than genetic algorithms through strategic, rather than random search requires initial learning phase and quality Oracle evaluations for optimal performance.
Good analysis. Let's continue. What's next? Future implications for AI-driven drug discovery?
The success of MolMem's memory-augmented approach signals broader adoption of episodic learning systems across drug discovery workflows. Future developments will likely integrate multimodal memory systems that combine molecular structure information with biological activity data, protein interaction patterns, and clinical outcome histories. Advanced versions may incorporate federated learning capabilities, allowing pharmaceutical companies to share optimization insights without revealing proprietary molecular structures. The integration of large language models trained on chemical literature could enhance semantic memory capabilities, providing context aware molecular optimization suggestions based on published research. Integration opportunities extend beyond molecular optimization to encompass entire drug discovery pipelines. MolMem's memory mechanisms could inform target identification by tracking successful molecular target interactions across projects. Combination with automated synthesis planning tools could create closed loop optimization systems that consider synthetic feasibility alongside molecular properties. Partnership with experimental automation platforms could enable real time Oracle updates from physical laboratory results rather than computational predictions alone. The broader implications suggest a shift toward more intelligent, memory enabled AI systems in pharmaceutical research. As Oracle evaluation costs continue rising with increasingly sophisticated property prediction requirements, memory augmented approaches will become essential for maintaining research productivity. The success of MolMem's agentic decision making may inspire similar approaches in other areas of drug discovery, from clinical trial design to pharmacovigilance monitoring, where expensive evaluations benefit from historical pattern recognition and strategic exploration strategies. Multimodal memory systems combining molecular, biological, and clinical data for comprehensive optimization, federated learning capabilities, enabling cross company knowledge sharing without proprietary data exposure, integration with automated synthesis, planning, and experimental platforms for closed loop discovery, expansion of memory augmented approaches to clinical trial design and pharmacovigilance applications.
Great analysis. So, what are the key takeaways from all of this?
MolMem reduces expensive Oracle evaluations by 60% through memory augmented reinforcement learning in molecular optimization workflows. Dual memory architecture combining episodic and semantic memory enables strategic molecular modifications without repeated expensive evaluations. Implementation requires Python 3.8 plus, molecular property prediction tools, and careful calibration of memory parameters and reward functions. System outperforms traditional template based and genetic algorithm approaches while maintaining molecular diversity and structural similarity.
And what market signals should our listeners watch for?
Sample efficiency becomes critical competitive advantage. As computational costs for molecular property prediction continue rising with more sophisticated models, pharmaceutical companies are prioritizing sample efficient optimization methods. MolMem's 60% reduction in Oracle calls directly translates to significant cost savings in early stage drug discovery, making memory augmented approaches essential for competitive research productivity. Companies adopting these methods can explore larger molecular spaces within existing computational budgets, potentially discovering novel drug candidates that competitors miss due to evaluation limitations. Memory augmented AI expands beyond molecular optimization. The success of MolMem's episodic and semantic memory systems signals broader adoption of memory augmented AI across drug discovery workflows. Pharmaceutical companies are recognizing that expensive evaluation scenarios, from clinical trials to regulatory submissions, benefit from historical pattern recognition and strategic decision making. This trend suggests significant investment opportunities in memory enhanced AI platforms that can reduce costs and improve outcomes across multiple stages of drug development, not just molecular optimization. Agentic AI systems gain traction in pharmaceutical research. MolMem's agentic reinforcement learning approach represents growing industry confidence in AI systems that make autonomous strategic decisions in drug discovery. Unlike traditional automated screening methods, agentic systems like MolMem actively choose which molecular modifications to explore based on learned patterns and optimization goals. This shift toward intelligent, self directed AI agents suggests pharmaceutical companies are moving beyond simple automation to embrace AI systems capable of scientific reasoning and strategic planning in research workflows.
What should developers and operators do with this information?
Implement MolMem for lead optimization campaigns when Oracle evaluation budgets exceed $50K per project and sample efficiency becomes cost critical. Deploy memory augmented optimization for novel target classes within 6 months of project initiation to maximize learning from early molecular evaluations. Integrate MolMem with existing computational chemistry workflows when teams perform more than 200 Oracle evaluations per optimization campaign. Scale memory augmented approaches to multi-parameter optimization projects requiring simultaneous improvement of 3+ molecular properties with conflicting requirements.
Thank you for listening to LeadAI.dev. Stay tuned for our next daily updates in this rapidly emerging market.